Welcome back, everyone. We have an update from Regen BioPharma, trades on the OTC Pink sheets under the symbol RGBP. It's a biotech company focused on developing innovative treatments using autologous cell therapies, RNA and DNA-based immunotherapy, and small molecules in the immune-oncology and autoimmune disease space. Let's welcome back President and CEO David Koos, as well as Harry Lander and Annette Marleau, Senior Scientific Consultants. Nice to see you guys again.
All right. It's good to be here.
All right, take it away, David.
Okay. So I'm excited about today because we finally get to talk about some results we've been waiting for. I know that everybody's been greatly anticipated getting an update on the results on our study. So I think at this point, I'm going to let Harry give an overview of the study. I'll jump in if I see there's points of clarification that maybe could be useful. And then after that, we're going to have an issue for Annette to address. And then as time permits, we'll take your questions at the end. So with that said, Harry.
Thanks, David, and welcome everybody. For those of you who have been following our story on this particular program called DuraCAR, you will likely recall that these are CAR T cells that have been designed, initially were designed to suppress one of the targets that we have focused on, NR2F6. And we've contracted with a couple of CROs to perform this over the last year. And just a quick, brief history recap for those of you who are new. These cells were initially thought that they would be very effective as cancer killers if we were able to suppress siRNA. It turns out that when we had our cells constructed, and it's a complicated construction, the cells did the opposite in terms of expressing NR2F6. So instead of suppressing NR2F6 mRNA levels went up quite high, which is to us a very useful finding potentially, because when you can modulate NR2F6 down or up with small molecules, and we have a small molecule agonist, something that activates NR2F6. So here we've done it genetically. And the idea is, well, okay, then if you're not activating the T cells, you're suppressing them. And sure enough, we designed some experiments to determine if we were suppressing the T cells or still activating them through some unknown mechanism. We got preliminary findings back about a week ago or so. And the company is going to redo the study in a much more in-depth way. However, they were able to transform the CAR. Normally, imagine if you start with 100 T cells and you want to transform them, you would expect to transform 80% of them, 70% of them. And in the CAR T space, normally if you transform 40% of them, you've done a good job. And those are the ones that are being infused in people. They're about 40%, 50% transformed. That is expressing the CAR antigen receptor. In their first set of experiments that they returned to us, they only were able to express it in about 15% of this particular batch. And so they're going to redo it. However, those cells were extremely effective as suppressing T cell activation. So we measured a series of markers on the outsides of T cells. And when you take normal T cells, unmodified, and you mix them with cancer cells, the T cells get activated. And there are proteins on the outside of the cell that show up on activated T cells, and you can measure them. So when we mixed our CAR T cells with this mixture of 15% modified T cells with the tumor cells, it dramatically suppressed certain activation markers, the IL-2 receptor, CD69, so certain receptors on the cell. So that is a very strong signal that these cells may indeed be immunosuppressant and may one day have a clinical utility. So we've
Terry, let me jump in for a second, just for a point of clarification. So basically, you're telling me we expected to find that we had a therapy potentially for curing cancer, and as it turns out, it looks like it's going to be effective potentially on curing autoimmune disease.
Yeah, initially we designed the cells to have anti-tumor activity, and when we saw those unexplained results, but we had to follow the science. We designed this last experiment to determine either way if it was going to suppress the immune system or activate and kill immune cells, and it did not activate or kill immune cells. It was actually a potent immunosuppressant.
Okay, cool. That I mean, it looks like it's kind of like we went to the car dealer to buy a Ford and ended up coming home with a Chevy.
Yes.
So now this next set of studies that they're going to do, they're approaching it a little bit differently, hoping that we're going to have a better outcome on the number of cells that are grown.
Yeah, it's not the number of cells, but it's the percent of cells that actually have our CAR, the number of DuraCAR cells in the mixture. So they expect to get it up to 30-40%, which would be double what they did last time, and we would expect to see even more dramatic results. But the results are already quite dramatic.
Okay. So how long do you think it's going to take for them to get the new set of cells?
I don't think it'll take that long. I would expect them to have the new set of cells done even next week. It's already been a week and a half or 2 weeks. So another week or so, and then they'll run those similar experiments. And there are many more experiments we've asked them to do, but until they get a good batch, they're going to hold off running the complete set of experiments. But to answer your question, let's give them a month to get a new set of results back.
But the results we have gotten back so far, you're very excited about.
I couldn't believe it. I mean, it really suggests that they're extremely potent because there were only 15% of those cells were transfected.
Now, in terms of the actual market sizes, would you say that it is actually more impressive that we potentially have an autoimmune drug than a cancer drug?
Oh yeah, the autoimmune space is very, very big, much bigger than the cancer space. All these patients need help, but having a cell therapy, there's a couple of now cell therapies, actually CAR T cells that are curing certain autoimmune disorders. There's I don't remember the name of the company, but there was a big deal that was just done, and they really have an amazing effect. So hopefully ours will be able to, sort of be part of that new generation of autoimmune targeted CAR T cells.
Okay, excellent. I'm trying to move things along here because I do have a question for Annette that I'd like her to address, and that is we had a shareholder that asked us if we are at a point where we can apply for grants, and if so, how much?
Sure. So absolutely. So I think, just in the interest of time, but to be comprehensive, I think it's very important. And so there are, in the pipeline, there are a lot of opportunities to apply. And I think that having, as the company does, a cleared IND and having other programs that are less advanced, grants can be used to advance those programs that are maybe a little bit less advanced and get exciting data akin to what Harry's was talking about for DuraCAR, and to press some of the other exciting programs forward, for which there's a lot of intellectual property already around some of the other products that would make them really sort of ready to explore in and really tap the rest of the value out by doing some preclinical and even clinical studies using, by applying to government agencies. And so I think that's absolutely important and, should be part of the plan, should be part of the program.
So let me jump in here. It seems like, well, we know that people would like to know which grants, how much, how quickly, all those sorts of things. It's similar to questions about material events that have not been publicly disclosed yet. So we can't sit here and tell you we're applying for this grant or that grant and how much the grant is for and get into specifics, because we don't want to jeopardize anything by doing so. But suffice it to say, we're very much interested in grants and what they can do for us and what they can do for our shareholders. Now, let's see. I got a question about how close are you to finalizing a joint venture licensing arrangements or funding? Once again, that's one of those questions that we can't answer right at this point because it hasn't been publicly disclosed yet, and we have to wait until an event takes place and we file an 8K on it before we can talk about things of that nature. Let's see. So I think we have quite a few things that in the pipeline that I believe in the near future we're going to be able to talk about, and I'm very excited about things that are in the pipeline right now. I know that doesn't cure the curiosity of all the shareholders that are wanting to know what's going on. But believe me, I think you can see that as things come up that are important, I try and be as transparent as I can and as the company can to make sure that you guys are up to speed, know what's going on, and feel like you can come to a venue like this and get as straight an answer as I can give you. Anna, do you have any other questions that have come in?
We sure do. We have lots of questions for you. So with the data the company has in place now, will it be enough to apply for a grant? Can you talk a little bit more of that? We've got lots of questions around that topic.
Yeah, I think that where we sit right now, we've got sufficient data to suggest that there's merit in applying for the types of grants that would be appropriate. But
and
as far as answering a bunch of questions about grants, we can't do that right now, or as you're in the process, and until you've completed the process, it could be deleterious to the outcome. In other words, we can't show you what's going in the chili. You got to wait till it's done and then sample it.
A follow-up question from Greg. How close are you to finalizing any joint venture, licensing agreements, or funding?
Yeah, those are things that we can't disclose publicly until we do it via an 8K. And that's once the event's taken place. I know it's frustrating. I know that people would like to know answers in real time. Unfortunately, in the legal world that we operate in, we can't always do that.
And Kanto asks, what specific clinical application for the Duracar cells are you going to see first?
Harry, you want to address that?
Yeah, well, so as we advance from this in vitro experiments, we'll move into animal studies, and that's when we're going to start to focus on which indications we'll want to focus on. And obviously, some of the top ones that one comes to mind is type 1 diabetes, huge market, arthritis. There are some very large markets that are still a lot of unmet medical need, a lot of need for these types of drugs.
And we have another question from Paris asking about the INDs. Why were the INDs from 2016 abandoned? Can you talk a little bit about that? And how long until possibly a buyout? What are your thoughts?
Well, we obviously can't talk about a buyout. As far as the IND goes, we are clear of the FDA, so it is an approved IND. We're in the process of going through the protocols on that and expect to have a further update in the near future. So we haven't abandoned it. So I think that we should have something positive for the near future.
And Kevin asks, are there any more planned reverse splits in the future?
No.
Okay. And give us a little bit of a timeline, if you can. And these can be your closing remarks, if you'd like, David.
Timeline. Well, when you're talking about medical technology and that, it's hard to pinpoint the timelines on things. I think that that in the Duracar situation, Harry's indicated that he expects to have some more results in about 30 days. That's safe to, yeah.
Yeah.
I think in the situation with the Heme Accelerate IND that we've cleared, I would expect to have an update for you on the next conference call.
Wonderful. Well, thank you so much, gentlemen. Always good with these updates, and we appreciate you coming on.
All right. Thank you very much.
Thanks,
Okay, everyone stay with us. We'll be back with Ideal Power at 3:40 Eastern.
Mm-hmm.